All right, we're going to continue here with the second lecture in the cell division unit, and we're going to be covering learning objectives 5 and 6. I've listed these learning objectives here for you in the video. Just please be aware that these are listed also for you on the page in the Canvas page. So I will be covering some of these questions that are listed under the learning objectives in a document that I put on the Canvas page. So learning objectives 5 and 6. What we're going to be covering is mitosis. Mitosis, if you remember, is part of the cell cycle in the M phase. So mitosis is conventionally broken into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. So if you notice here, we have a pretty large diagram here showing each of these phases as we go through. Here's interphase before it starts, before it starts into the actual mitosis. So interphase, prophase, prometaphase, then metaphase, and then anaphase, and telophase. So these are the stages of mitosis that we will be going over in some detail. So one thing that I wanted to show you though is that if you are looking in your textbook, this is a really important figure for you to look at. So let me just show you in the e-text here. In figure 12.7, you'll see the exact same diagrams that I'm getting ready to use. So G2 of interphase going into prophase, prometaphase. This is the diagram that I would study. There are extensive descriptions for what's going on in each stage that are showing up below there. So these are actual photomicrographs showing you what it would look like in a real cell. It's a little bit easier to see when we just look at the drawing though. So spend some time in this figure looking at the actual figure, looking at the descriptions of what's going on, paying attention to what you see the chromosomes doing and what you see these cytoplasmic elements such as this structure called the spindle doing, and these structures called centrosomes. Now what I would ask you to do is be able to draw these cells in each one of these stages from memory. For example, this is showing 1, 2, 3, 4, 5, 6 chromosomes. Maybe what if you had to draw one with only 3? Would you be able to take it through these stages and show what it looks like with 3 chromosomes going through mitosis? So now that I've shown you that, I'm going to go back to the PowerPoint and we're going to look at each one of these stages. So the first stage is prophase. However, before we get to prophase, we back up a little bit and see what it looked like before it went into mitosis by looking at G2 of interphase. We always notice what's going on in the nucleus and what are some specific things going on out in the cytoplasm. And in the cytoplasm, we're going to look at the centrosomes. The centrosomes are going to be a structure that the spindle is going to be built from. So we'll be following that spindle as we go along. All right, so we see here, first of all, our chromosomes in the nucleus are not visible as individual chromosomes. At this stage, we call it chromatin. But they are duplicated chromosomes, but they're not condensed. Going on into prophase, we do see that they condense. Now they're visible as individual chromosomes. We see the nuclear envelope. We see that the nucleolus has disappeared. Here was originally and how it's disappeared. We see the beginning of the mitotic spindle, which is emanating from these centrosomes. In prometaphase, we see the nuclear envelope start to fragment and break apart. We see the mitotic spindle practically complete here, and we see our chromosomes. One important thing about the chromosomes is this structure right here. It's colored black in this case. It's called a kinetochore. A kinetochore is the region, it's a protein that's in the centromere region, and it's where these microtubules from the spindle attach. It's very important that all of these kinetochores are attached to a microtubule. In this case, we've caught it in the middle of the process, so all of them have not been attached yet, but it is in progress. Now, these are called kinetochore microtubules if they attach to the kinetochore. There are other kinds which do not attach, and these are going to be involved in lengthening of the cell as it goes through division. Going on to the next phase is metaphase. Metaphase starts with an M. I always just think middle. They line up along the middle. So you see your chromosomes lined up along an imaginary plate. In each case here, now we have all of our kinetochores attached to a kinetochore microtubule. Remember that this structure right here is called a duplicated chromosome, and it's composed of 2 sister chromatids. So each sister chromatid has a kinetochore. They both have to be attached to the microtubule. All right, at anaphase, what happens is these sister chromatids are released from each other. So one of them is pulled one way and one sister chromatid is pulled the other way. There's a protein called cohesin that was holding them together, and that is cleaved, allowing them to be separated from each other. Notice that they're now called daughter chromosomes. Originally, when they were attached and held together, they were called sister chromatids. Now they're daughter chromosomes. Moving into the last phase called telophase. Telophase is formally defined as when at the end of telophase, 2 new nuclei have reformed. So this is in the process here. We're having our nuclear envelope reforming, the nucleolus is reforming, and the spindle is disappearing. But if you'll notice, at the same time, the process of cytokinesis is overlapping with telophase. So cytokinesis is the pinching apart, in this case, of these 2 new cells, or the division of the cytoplasm. All right. Okay, well, this is a video and I'm not able to get it started, but it's a video showing, you can tell how you do see these, I know how to do it. Okay, we can watch it by doing that. Let's see, let me share the screen with you again. All right, we can watch it from here.
Watch the formation of the nuclear envelopes in a dividing embryo of C. elegans during 2 rounds of mitosis. The image on the left shows the location of yellow fluorescent protein labeled lamin, the nuclear membrane protein. Notice how the nuclear membranes disappear and reform in each cycle of division. Credit, Mitosis, European Molecular Biology Laboratory, Germany.
Okay, so we were seeing the formation of the nuclear envelope there. In this next one, we're looking at all of these stages of mitosis occurring. So you'll see the chromosomes line up along an imaginary plate, and then you'll see them separate. Okay, and the separation occurred really quickly there. And now you're seeing cytokinesis occur. All right, let's go on to the next part here where I wanted to look in a little bit more detail about what's going on. Look at this a little bit more, about what's going on here at this mitotic spindle when we have all these chromosomes lined up during metaphase. Here's what it would look like in an actual cell. So I can label some of these structures. We can label, here's our mitotic spindle. Here are, of course, our kinetochores, these black structures here. We have our non-kinetochore microtubules that don't actually attach to a kinetochore. And then we have our duplicated chromosome, which is composed of 2 sister chromatids. Now, some other names for it. We see this structure up here. It's just the structure that's seen. It sort of looks like a little starburst up here, so it's called the aster. Aster really just means star. This is just a description of the things that are going on at the mitotic spindle. A mitotic spindle is a structure made of microtubules that controls chromosome movement during mitosis. In animal cells, this assembly begins at the centrosome. The centrosome is a microtubule organizing center. So the centrosome replicates itself during interphase. And so there are 2 centrosomes at the beginning of prophase, and they migrate to opposite ends of the cell, during prophase and prometaphase. There's an aster which extends from each centrosome. The spindle itself includes the centrosomes, the spindle microtubules, and the asters. All right, so what happens during prometaphase is some of the spindle microtubules attach to the kinetochores of chromosomes and begin to move the chromosomes. And what the kinetochores are protein complexes that are associated at the centromere region. At metaphase, the chromosomes are all lined up at the metaphase plate, which is a plane midway between the 2 poles. Let's just skip that one for now. In anaphase, when the chromosomes are separated, the cohesins are cleaved by an enzyme, and that enzyme is called separase, to separate the sister chromatids. The sister chromatids separate and move along the kinetochore microtubules toward opposite ends of the cell. Non-kinetochore microtubules from opposite poles overlap and push against each other to elongate the cell. At the end of anaphase, the duplicate groups of chromosomes have arrived at opposite ends of the elongated cell. Cytokinesis begins during anaphase or telophase, and the spindle eventually disassembles. So here's a diagram here of some just shapes that I have put on here. Here's my imaginary cell. Here's a cell that has 2 chromosomes. They're aligned along an imaginary plate at metaphase. The reason I put this here is to suggest to you that it would be helpful for you to be able to make your own drawings. I didn't include the spindle here, but that would have been helpful. Be able to make your own drawings of each of these phases. And if drawing is difficult, you don't want to do a lot of drawing, what I often do is cut out these shapes. So this would be 1 duplicated chromosome, but it would be composed of 2 sister chromatids. So this would be a sister chromatid, this would be a sister chromatid. So then it's easy to move these shapes along and imagine where do they align, how do they separate, where do the microtubules attach. Remember, we're going to have a microtubule attaching at each this would be our kinetochore region here at the centromere. All right, so that's just a suggestion for you to help you study. Let's look a little bit more at cytokinesis, and then we'll be finished with this video. In animal cells, cytokinesis occurs by a process called cleavage. In plant cells, we call it cell plate formation. So let's look at the difference between those 2. In animal cells, they literally, not literally, but it appears as if they're pinching apart. Here is a structure called a cleavage furrow. It's just where you see that pinching in area. What's happening there is a cytoskeleton component called microfilaments that's just contracting there. It contracts until these daughter cells are eventually pinched apart into 2 daughter cells. In the plant cell, however, the rigid cell wall does not allow them to be pinched apart. Instead, new cell wall material is laid down between the 2 new nuclei. So where this cell wall comes from is vesicles budding off of the Golgi containing the proteins and cellulose to make the new cell wall. These vesicles eventually fuse in a cell plate, and when they go all the way across, this is called the new cell wall. So this process is called cell plate formation. Contrast that to cleavage, which occurs in animal cells. All right, and that's the end of this portion of the discussion of cell division. Thank you.